Yeah, so I'm Ben Dichter, the founder of CatalystNeuro, and I'll be talking about open neurophysiology data for education. So the data that we collect, I'm sure I don't have to sell you all on open science, but it's very valuable. It takes money and time, and we use animal lives, so it's expensive in all of those different ways. And we know that it can be used to answer many different scientific questions, can be used for education, but it requires infrastructure to standardize and share this data. We know that it can have this enormous potential for research and education, but it requires both technological and social changes to actually make that happen. So this is really the core mission behind a company that I founded about 7 years ago called CatalystNeuro. So we're an international team of research software engineers, and we connect data producers, tool builders, and data users in neurophysiology research. So the main technologies that we specialize in are NWB and DANDY. So NWB stands for Neurodata Without Borders, and this is a data standard for neurophysiology that allows the data to be standardized and shared across groups all over the world. And the DANDY archive, which is an archive and collaboration space for neurophysiology and behavioral data. So people often ask me what's the difference between NWB and DANDY. I like to say NWB is kind of like MP4. I don't know if you guys all remember earlier in the the video streaming days, there were tons of different file types. You didn't know how to read them in on different computers, but MP4 kind of solved that problem. Now you can easily share a file with someone else, and you can trust that they have software that knows how to read it. So that's what NWB does for neurophysiology. And then YouTube is a place where you can upload these files for free, download, search, use them for your own purposes. So that's kind of what DANDY is doing for neurophysiology. So NWB, the goal of NWB is to package all of the neurophysiology data with the metadata necessary for reanalysis. So this, of course, includes the neural activity on the left, your actual measurements from your extracellular, intracellular, or calcium imaging experiments. But it also contains metadata about the data acquisition system, the experiment design, the experiment subjects, the behavior of those subjects, and it also contains tools for building extensions to NWB, so you can handle types of data maybe for new types of analysis or acquisition systems. So it's been a challenge getting the community to adopt these open standards, and I really like to think about kind of the energy barrier of open science and ways that we can influence this energy barrier. So one way to do it is through what I would call sticks, funding ultimatums, right? And this is something that the NIH has already started to do. They said, hey, if you publish a paper, you have to share the data that's corresponding to that paper. That started a few years ago, which is really powerful, but it can't be used in isolation, right? If all we did was say that, we would just get people sharing a bunch of junk, right? So what we also need to do is offer carrots. We need to offer maybe analysis and visualization tools that make sharing that data advantageous to the researcher, right? Now they're incentivized to actually do this because they can access these powerful tools. And then the third way is through guidance, right? Through lowering that energy barrier itself, through training, for instance, documentation, tutorials, hackathons, through working directly with neuroscience labs and providing custom solutions, and through building and distributing open source tools that make this easier for them. So the guidance area is where my company is mostly focused on. We're part of the NWB and DANDY effort, and we really focus on working directly with labs, understanding their bottlenecks, and trying to help them with that. And we've been actually really quite successful, if I do say so myself, and getting data onto the archive. This is a plot that I'm very proud of now over 375 data sets in NWB, publicly available, published on the DANDY archive. And this comprises, it's just coincidentally, the number of terabytes is almost exactly the number of data sets. So we have over 375 terabytes of neurophysiology data on the archive. This is a lot of mouse and rat data, but it's certainly not limited to that. We also have a lot of human and monkey data, zebrafish, fruit fly, and then a few other model organisms, and also a lot of really non-model organisms. So you can publish all sorts of data on this archive. And a lot of it is extracellular electrophysiology data, but it also includes a lot of calcium imaging and intracellular electrophysiology. And since I'm here in the Allen Institute, I wanted to emphasize that one of the most impactful users, probably the most impactful user of the Dandy archive and NWB is the Allen Institute, which has been a huge champion of open data and of this particular infrastructure from the beginning. They have open scope data sets. So this is a program that has published 10 data sets, calcium imaging and neuropixel electrophysiology. There's the BICAN program. There's dating all the way back to the Brain Observatory, which was really one of the first publicly available neuropixel and high-density 2-photon data sets, and now has over 100 citations of people reusing this data. So I would say the Brain Observatory has received the most reuse, but it's actually it's older data at this point, right? So we actually have even more sophisticated data. So I think the future is really bright for data reuse. So talking more about the open scope program. So this is a program where members of the neuroscience community can propose experiments for the Allen Institute to run. And then they choose across those proposals and they pick their favorites, and then they use the Allen Institute infrastructure to run that, and then package the data in a way that is intended to be reused, right? So the proposers of the experiment get exclusive access to that data for a short amount of time, and then the whole neuroscience community has access to it. And now they've already published 10, I think maybe 11 by this point. They were working on an 11th when I created this slide. And it's about half calcium imaging and half electrophysiology. Lots of really fantastic experiments, mostly around visual processing and predictive coding, and kind of the interaction between bottom-up and top-down processing. So one of the reasons that I want to emphasize these data sets, one is because they're really great data sets, but also because they come with this open scope data book. So this is an electronic, openly available, interactive neuroscience test book with runnable tutorials. And it teaches you how to use the open scope data that's stored on Dandy, freely available in NWB. So you start with these basics: downloading, reading, exploring NWB files, how to stream them directly from the archive, and how to get and process the metadata. Then you get into a bit more advanced visualizing the behavioral, the calcium imaging, electrophysiology, both raw and processed, basically how to create those standard visualizations based on NWB data in Dandy. Then you get to the first order analyses. You can see down here, I don't know if you can see it, but it says showing receptive fields, identifying optotagging units. It actually, so this is a great resource. I'll admit it gets pretty advanced pretty fast. And if you continue to go down, there's like Zebra, which is like a neural network for dimensionality reduction, which like there's no soft intro, it's just like straight into it. So I would say I really love this resource. It was not designed for undergraduates. So I just want to be honest about kind of where we are and like why I'm here, right? Is like I want to work with you all to create resources that are more appropriate for undergraduates. Because we have all sorts of data. So that was data from the Allen Institute, but that's actually a small subset of the data on the archive. So it's really impossible to summarize this, but oh, by the way, I see people taking pictures, which you're welcome to do, but also that URL down there on the bottom right is all of the slides with links. So if you're interested in these slides, check out that URL. Okay, so yeah, just a brief summary of the data on the archive. I mean, we have data that's about hippocampus and navigation. I think we have like 61 data sets from like highly influential labs, the Buzaki lab, the Frank lab, the Giacomo lab. We have sensory processing, vision, hearing, smell, touch, dozens of data sets having to do with sensory processing. Evidence accumulation and decision making. We have the data set, the brain-wide map from the International Brain Lab, which is like a 22 lab collaboration. Working memory, motor control, roles of different neurotransmitters. We're working with groups that are collecting data from animal models of Parkinson's and autism. So we've got tons of different types of data spanning lots of different topics. So let's say you wanted to actually explore these data sets. So one of the really great ways of exploring the Dandy archive is through NeuroSift. So this is an it creates intelligent interactive visualizations for NWB data, and it runs in browser. You don't need to install anything or run any external services. It streams data directly from the archive and works with the Dandy archive. So I tried these earlier and the URL was blocked, but we'll see. One of the really great things about this is that it just works, but it only works if I can actually access the URL. But so I want to talk about one data set, which is this one I'm showing here on the left, was one of the data sets contributed by the Moser lab that was actually cited in their Nobel Prize announcement. So this is one of the key data sets where they found grid cells in the entorhinal cortex. And we worked with them to help them get their data on the archive. It's now freely available. But let's say you wanted to actually explore that. Okay. This is a link to the data on the archive. You can download this data, by the way, without you don't even have to register for Dandy. So you can just click download. You can do a simple pip install. You can go into these files. I'll just go to one file here. You can just download this, and that will get the NWB data on your computer, and you can start using it. But one of the really great ways to explore this is through NeuroSift. And it works! Okay, perfect. So, you can't see my screen. Thank you. Okay.
I would share again.
Yeah, I'm just not used to Teams. I don't know how to share.
Oh, the share button top right.
Yeah, top. Yeah, the lifter.
I don't see that on this computer. Well, I want to share the. Sorry, I want to share the. Yeah, but all right. Well, we might have to skip over this. Sorry, guys. Oh, thank you.
I do work for Teams, if that helps.
Well, how do I share this screen? Share sound optimized layout. I don't.
Oh yeah. Stop sharing.
Okay.
And then share.
This is gonna be worth it, guys. I promise.
And then window. And we probably screen is better.
Yeah.
Specific screen.
All right. Okay. Here we go. Thank you so much. All right. So, really quickly, if you go to dandyarchive.org, I'm just going to type in Moser here. This is the data set that I was talking about. You can look at the metadata corresponding to this. You can go into files, and you can just download data right here. You don't even need to sign in. You can just click this, and it'll download it. Whoops, that actually downloaded it. All right. Well, now you have NWB data. Or you can use NeuroSift, where you can explore this without doing any coding. So I'm going to look at a few different data streams here. I'm just going to click this and this and this. So in this data set, we have a rat who has electrophysiology being recorded directly from his brain. From that, there is spike sorted data. Oh, this failed to fetch. That's interesting. Okay. Well, here's the raster plot of the units that have been extracted. And here's the XY position of the animal over time, and here's X versus Y. So this is pulling data directly from the archive and giving you a view to interactively explore this data. All right. And then let's say you're like, okay, yes, these are the types of data streams I'm interested in. Now I want to actually analyze them. So you can close this, and there's a tab here, Python usage. And here's all the code. So you can copy this and paste it directly into your Jupyter notebook or anything. And this will actually is all the code you need to stream the data directly from the Dandy archive into your analysis script. So this will give you the the electrode location and position, the unit spike times, the position of the animal over time, like everything you need. Okay, now I need to go back to the slides. All right, we're good. And then, okay. I don't quite have time for the peristimulus time histogram on the IBL data set, but here's the data set from the IBL where animals are shown images on the left and right, and they have to make a decision about whether the image was shown on the left or right. Using NeuroSift, we can take that data and reshape it to create peristimulus time histograms, aligning to these task events and showing the response of different neural activity based on different contrast levels of those images. So all of this can be done without any coding, just with interactive dashboards that stream directly with the Dandy archive. We also have the Dandy Hub, which you can use for free. It's a computational resource where you can run different programming languages. We have a collection of community notebooks that show you how to read and analyze data from many different data sets. These are like community contributed notebooks showing, so for instance, let's say this one, you can pull up notebooks showing you how to read and analyze data from all these different data sets. So you can use these and you can also contribute your own. Let me go back here. Okay. We're exploring AI tools for summarizing data sets, intelligently searching the archive, analyzing data sets. I want to give you a really quick sneak preview of this. So this is that Moser Lab data set that I was showing you before, and here's a chat about it. So you can say, tell me about this data set, and it will think about it and give you a response based on the metadata there. You can also type in and have a conversation with this, asking how to actually analyze it, or you can say auto ask, and it will just kind of continually kind of have a conversation with itself that you can kind of jump into. This is actually writing code, which it can then run. And if you're interested in that, I will happily kind of show you that later. Okay, I have a few minutes left. One of the really valuable teaching resources, so this is something created by Ashley, actually, and this is an educational resource that is more, I think, appropriate for undergraduate education. It's teaching and learning with NWB datasets. The URL is nwb4edu. It's interactive online textbook for educators and students and researchers to learn about data analysis using NWB and Dandy. It has lessons in PyNWB, which tells you about NWB data structure, extracellular recording analysis. It has lessons about the Allen SDK to get data and metadata specific to the Allen Institute. And it also has the section on the greatest hits in neuroscience, recreating published figures from key findings with NWB data. So right now there are just two of these. One is how does the brain encode space, which actually uses that Moser Lab data set. And the other is how do populations of neurons encode movement. So this shows like directional reach tuning for like a monkey reaching task. And what's really cool about these is they actually are really designed for educational purposes. So there are spots in there where there's like left blank and say, okay, here's where you calculate the rate map and you have to code it yourself. So this is one of the things that I think we maybe need more of. Like I think we have all these notebooks that show you how to do things, but most of them are not really designed for pedagogical purposes, in particular for undergraduates where you want these like interactive types of exercises. And so that's where I'd really love to work with you all. I mean, I think this is a really great resource, but these are just two of the greatest hits, and we could easily have 10 or 20 or more. And I'd really love to like iterate with you to understand how best to deliver this type of content. So thinking back to this kind of energy barrier and how can we address this specifically for undergraduates, I really want to be able to create and disseminate educational resources, and I think maybe this is the group to help us do that. We've been doing a lot of outreach, but it's been primarily focused on senior graduate students, and we have come to realize that that's really not ideal. We would really love for people to go into grad school understanding these resources, maybe not being an expert, but having in their mind, oh, if I have a scientific question, maybe I can run a query on open data sets instead of collecting my own data. Just like if we can kind of get that across and demonstrate to them that this is possible, that would be a huge step forward. So how do we actually accomplish this? Through outreach, talking to educators, maybe talking to student organizations, maybe other groups. I'd love to hear your thoughts on that. How can we incentivize people to actually work on this? Are there funding mechanisms that would facilitate this? Maybe we could have some kind of project competition. Maybe they would have prize money involved. I think we need more venues for undergraduate scientists to give presentations. I've heard a lot about that here, which I think is awesome, and I think we just need more of that. Yeah, I want to thank the funders for support, NIH, Kavli, Simons, Michael J. Fox, and the Dandy and NWB teams.